Last lecture, we talked about regulating access to the naked DNA so that transcription can occur. And that's at the level of chromatin and sort of the histone modifications and the cytosine modifications that can occur on the DNA. But now that we've got the naked DNA available, we can look at the regulation of actually transcription. And so transcription is regulated by special transcription factors that are different than the transcription factors we talked about before. So let's talk about the DNA regions involved in transcription regulation. So the regions in the DNA that are play a role here is we have the basal promoter or the core promoter, which we talked about before. This is required. This is not, it's required for transcription, but it's not part of the sort of regulatory process. And remember, it's directly upstream of the coding region. And in eukaryotes, it's got that very special Tata box. So it's got the Tata box where basal transcription factors bind. So where the basal transcription factors or general transcription factors bind and recruit RNA polymerase II. There are additional regions involved though. So this we've talked about before. We talked about that basal promoter before. And sometimes these are considered part of the basal promoter, but there are promoter, sometimes you will hear promoter proximal elements. Proximal, proximity, nearby. So these are DNA sequences near the promoter that are also involved in sort of involved in regulating transcription. So they're near the promoter, and sometimes we refer to sometimes the GC box that we discussed before and the CCAAT box are considered promoter proximal elements. So now what other factors do we have here? We're talking about at the level of transcription. Here we've got our +1 transcription start site. We've got a couple of exons and intron. We have the promoter, the core promoter with the Tata box. Promoter proximal elements are upstream of the promoter, but they're still kind of linearly nearby. You know, and this can span from -35 to 300. These additional elements outside of there, enhancers are a type of regulatory region on the DNA that play a role in activating transcription. So we can think of enhancers as activator binding sites. And they are short DNA sequences. So what do I mean by short? You know, less than, you know, anywhere from 4 to less than 20 base pairs in length. So they're short DNA sequences, and there are hundreds of different enhancers. Now, while promoter proximal elements and promoters are directly upstream of the transcription unit, enhancers can be some distant. So they can be distant from the promoter and they can be upstream. So they can be thousands of base pairs away. They can also be downstream. Sometimes they can even be in the transcription unit itself, in the introns of the transcription units, and they can be in multiple copies. So these are activator binding sites enhancers. The counterpoint to the enhancers, and they often overlap the enhancers, are called silencers. Oops. Silencers are, you can think about these as repressor binding sites. And they often overlap. So they've got the same characteristics as enhancers, and they often overlap enhancers. So these are the DNA regions. So what binds to these regions and how do they control transcription? So let's talk about the regulatory proteins. Oh, here's some proximal elements. Here are some enhancers, also called distal, meaning distant control elements. Here's our core promoter with the Tata box in here. So what is it that binds to these different regulatory regions? So let's refer to these as the regulatory proteins involved in transcription regulation. Well, we know that basal transcription factors, whoops, excuse me here, this is a little one. Basal transcription factors are required in RNA polymerase II bind the core promoter and they're required for transcription, right? So they have to be there. In addition to those, though, we have activator transcription factors. Activator transcription factors are specialized or special transcription factors, which are proteins that bind to enhancers and upregulate transcription. There are hundreds of different of these specialized transcription factors. There are hundreds of different transcription factors, and each transcription factor binds its unique enhancer sequence. So what do I mean by that? Each binds its enhancer sequence. So let's take a look here. I've got a table. So here are some different transcription factors: SP1, AP1, ATF, CMYC. This is involved in a lot of cell growth pathways. NF1, OCT1, which is involved in stem cell regulation. So these are different. These are the proteins. These are the specialized transcription factors, so TFs. This tells you a little bit about the protein structure, which we don't care about so much, but here are the recognition sequences. So these are the enhancers to which each of these bind. So these are the enhancer sequences to which each of these bind. Let's look at CMYC. It binds CAC, GTG. That's, again, the language of the coding strand. And that is the sequence to where CMYC binds. That is the enhancer region to which CMYC binds. So we've got our activator transcription factors that bind to their unique enhancer sequence. And then repressor proteins bind to silencers, unique silencer sequences, their own unique silencer sequence, and block or downregulate transcription. They do that by blocking activator binding to the enhancer site. Here we can take a look at how this actually occurs. Here we've got our promoter, or core promoter with polymerase. Here's are a couple of enhancers with activating transcription factors that bind. So the activating transcription factors bind to the enhancers, they loop around, and they essentially like say, tag, you're it, and they cause the transcription to occur. Let's see if this link is working.
Successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TATA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers, scattered around the chromosome, can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.
So that is sort of the mechanics of how activating transcription factors and enhancers work to regulate transcription. But here's kind of the magic. How do these different cell types express different genes? What genes are expressed when? The key to that is that different genes, so those 20,000 plus different genes in the human genome, different genes have different regulatory elements. And this is just another term for silencers and enhancers. So different genes have different regulatory elements and therefore are activated or turned on or upregulated. They're activated by different transcription factors. And here I'm using transcription factors, talking about those specialized activating transcription factors. So again, here's another depiction showing our core promoter. These are cofactors, proximal elements binding cofactors. And then here's our activating transcription factors. Here's showing the silencer blocking the activator from binding. Here are those different ones again. Now here, let's imagine we've got a liver cell and we've got a lens of your eye cell. Okay, so these cells do very, very different things, right? They do very different things in your cellular physiology. So, but the genes that are in each of these cells are exactly the same. So how do they do their different things? Because it's the proteins that do stuff. So how do you get different protein expression? Well, when we're talking about at the level of transcription factors and enhancers, let's imagine two different genes. Here's an albumin gene. This is a gene that codes for a protein that's in abundance in the serum. It's involved in detoxification sort of processes. And then here's a crystalline gene. So the crystalline gene codes for a protein that's involved in vision. Okay, so we've got albumin and crystalline. They both have promoters, right? So Tata box would be in here. They both have proximal elements, but they have different associated enhancers. And so just by, in a cartoon form, the albumin gene has an enhancer that's yellow and gray and kind of this red color. And then the crystalline gene has orange and gray. So they share gray, but they have different other enhancers involved. So let's see, if we're in the liver cell nucleus and we look at transcription factors that are being expressed in the liver cell nucleus or being activated in the liver cell nucleus, we see transcription factor red, orange, yellow, and gray. So they bind to the enhancer sequence and they fully turn on transcription of albumin. But look at, there is no pink and there is no orange transcription factor. So the crystalline gene is not expressed. Now, in contrast, if we look at the lens of the eyeball cells, what we see here is we have a different set of activated transcription factors in the nucleus, and therefore we get expression of the crystalline gene and not of the albumin. So what turns these on, or what expresses these different transcription factors in the different cell types? Here, I'm going to do this by example. So transcription factor activity. So this means either binding to the DNA or it could be expression, is regulated by the cellular environment. And we're going to look at a specific example, a favorite of mine, because this is my graduate school dissertation, was on the effects of glucocorticoids on your immune system. So let's use the example of glucocorticoids. Should ring a bell. We're going to shorthand this, GCs. So what are glucocorticoids? They are steroid hormones. There are steroid hormones that regulate carbohydrate metabolism and immune function. And they increase, blood serum concentrations of carbohydrates increase with stress. So they're referred to as our stress hormone. So glucocorticoids increase with stress. Now, stress is not always a bad thing. Stress could be something like going for a jog or working out. And in that case, glucocorticoids would come up and come back down and actually has a positive influence on physiology. But if you have like a traumatic stress or chronic stress, glucocorticoid levels can go up and stay up, and then they can cause dysfunction. So just as a general here, here is where the glucocorticoids are produced. So under stress, the thalamus, the hypothalamus, triggers the synthesis and the release of cortisol from the adrenal glands. So stress triggers sort of upregulation of one of the glucocorticoids we'll call cortisol. There are different versions. Cortisol is one of the glucocorticoids. So now we have higher concentrations of cortisol in this blood serum. So how does that signal to our cells? Let's imagine a cell here, the nucleus of a cell. Now our levels of glucocorticoid steroid hormones have just gone up. If you remember what steroids are, they belong to the class of lipid macromolecules. And so they have some ability to diffuse through the cell. So a signal is not a signal unless it has something to receive it. Inside the cytosol of cells is the glucocorticoid receptor. So this is a receptor that's going to receive the signal. Glucocorticoids can diffuse into the cell, bind to the receptor. It's actually going to cause a dimerization to occur. So two of them are going to bind together. That causes it to enter the nucleus. So the glucocorticoids diffuse into the cell. They bind their receptor. So they bind to and activate the glucocorticoid receptor. Now, the glucocorticoid receptor, when it's activated, it translocates to the nucleus. So the activated glucocorticoid receptor translocates to the nucleus of the cell. Now get this. This is very, very cool. What's in the nucleus? Let me try to make a little helix in here. The glucocorticoid receptor is a transcription factor. It acts as a transcription factor itself. The active glucocorticoid receptor acts as a transcription factor. The glucocorticoid receptor binds to its enhancer, which is called a glucocorticoid response element, or GRE. So anywhere that a GRE exists and it's available, and the naked DNA is available, the glucocorticoid receptor is going to bind, it's going to act as a transcription factor, and it's going to upregulate genes that have the glucocorticoid response element. Now, not all genes have glucocorticoid response elements. We know of approximately 2,000 genes that have glucocorticoid response elements. That means approximately 2,000 different genes can respond to upregulation of glucocorticoids. Now, those are genes that code for proteins that are involved in carbohydrate metabolism and immune cell function. And so it's really kind of this beautiful system, right? Here's another depiction. Here's like my drawing. You can actually see in here the glucocorticoid response element sequence on the DNA. And here is an animation.
Together, we lost 40 pounds on Nutrisystem, but I really recommend.
Steroid hormones are not water soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.